Chapter 3, The Cellular Level of Organization, will be delivered by video lecture. Please watch the video as you would attend class, taking any notes that you need. Also remember to check Wiley Plus for homework assignments, and also check Wiley Plus for quizzes that may become due. Thank you, and I hope you find this informative. Chapter 3, The Cellular Level of Organization. So we've been moving our way up very slowly from a general introduction to human anatomy and physiology, to the chemical level of organization, and now to the cellular level of organization. We're going to go through the parts of the cell, beginning with the plasma membrane, moving to the cytoplasm, and then discussing the nucleus. So first let's talk about the generalized cell. The generalized cell has these three main portions: the plasma membrane, which forms the cell's outer boundary. We already looked a little bit when we were in chemistry about some of the components of the plasma membrane, but we're going to be getting into it in a little more detail. So the plasma membrane separates the cell's internal environment from the outside environment. It essentially separates the intracellular fluid from the interstitial fluid. Remember, the body has three fluid compartments: the intracellular fluid, the interstitial fluid, and the blood plasma. The plasma membrane will control the movement of materials from the interstitial fluid to the intracellular fluid. It is a selective barrier. In fact, we often say that the cell membrane is selectively permeable. In addition to controlling what enters and what exits the cell, we also, in the cell, say that it plays a role in cellular communication. So the cell will allow, the cell membrane will allow one cell to speak to another. We'll look in a lot more detail about these structures of the plasma membrane that allows it to accomplish these various goals. Next, we have the cytoplasm. So the cytoplasm is all the material in the cell between that plasma membrane and the nucleus. It includes two main portions that are mixed together. First, the cytosol is the fluid portion. This is mostly water. This is specifically what we refer to as the intracellular fluid. Mixed within the cytosol, we have all of the various organelles. Organelles are subcellular structures, so on their own, an organelle would not be considered alive. But the name literally means little organ, and just like an organ has a characteristic shape and function, organelles each have very characteristic shapes and specific functions. Organelles have the same shape and function, whether we're talking about human cells or frog cells or plant cells. The cells have the same types of organelles. And finally, we come to our next major area of the cell nucleus. Many people consider the nucleus to be a very large organelle, but it's a large enough of an organelle that we give it its own structure within the cell. It contains your DNA. The DNA is your genetic material. DNA is organized into chromosomes. Now, there are a couple of different definitions of the term chromosome. One definition of chromosome that you see here is that it is a single molecule of DNA and associated proteins. Humans have 46 chromosomes. Chromosomes in humans are paired. So they will be paired structures, and there are 23 pairs of chromosomes. So it's a single molecule of DNA So you could also say that a human would have 46 molecules of DNA and some associated chromosomes. The chromosomes will contain the hereditary units called genes. Genes are essentially a location on an individual chromosome. Another definition that you often see for chromosome, we will talk about in more detail when we talk about cell division. A chromosome could also refer to a structural form of the DNA. When a cell is not actively dividing, the DNA of the chromosome is spread out. It's found as long, thin strands. When a cell gets ready to divide, the DNA condenses. That means it coils up around many of the proteins and forms very distinct structures that are smaller, and those may also be called chromosomes. So it's important to figure out what type of chromosome a person is talking about from the context. So here's our generalized body cell. You can see that on the surface we do have this plasma membrane. So we have the plasma membrane out here on the surface. It's going to separate what is inside the cell from what is outside of the cell. We have all of the material in between. So all of this material in between right here, that is the cytoplasm. And then finally we have the nucleus. So here you can see the nucleus outlined in red, a very large organelle, but a very important organelle, important enough that we give it its own level of organization within the cell. So let's go back to the plasma membrane and examine it in a little more detail. It is a flexible but sturdy barrier. When we talk about the plasma membrane, we often use the term fluid mosaic model of the cell membrane. This term refers to the fluid portion of the membrane, which would be the phospholipids, and a mosaic portion, literally looking like mosaic tiles, those would be the protein molecules. So we have an arrangement of protein molecules within a sea of lipids. The lipids are primarily going to be a barrier. Some molecules will be able to pass through the lipid layer, and the lipid generally will consist of two different forms. As you recall, we have the phospholipids, and we also have a very important molecule known as cholesterol. These are both going to be very important components of the lipid portion of the plasma membrane. Phospholipids and cholesterol, both are lipids, and lipids hate water. So any molecule that is very polar, such as an ion, cannot pass through this lipid barrier. Also, molecules that are too large, like starch or glycogen, which are both examples of polysaccharides. Although they are attracted to water, they have only slight charges, but they are just structurally too large to get through the phospholipid bilayer and the cholesterol. So the lipid portion is a barrier. Some molecules will be able to pass through. Examples of molecules that can easily pass through the phospholipid bilayer would include molecular oxygen, the oxygen that you breathe. Carbon dioxide can pass through the phospholipid bilayer, and also water, to a limited degree, can also pass directly through the phospholipid bilayer. The protein portion of the plasma membrane is how we say that the plasma membrane is selectively permeable. Proteins act as gatekeepers. They will form channels or carrier structures to allow certain molecules and certain ions to pass through. But just like a gate, sometimes the proteins could be open, sometimes they could be shut. So the cell can change whether it will allow ions to pass through or whether it will stop them. So here we see the structure of the phospholipid bilayer, the cholesterol, and the proteins. So we can see our phospholipid bilayer, and remember, when we draw a phospholipid bilayer, we draw the phospholipid with the polar phosphate head and the fatty acid tails. Remember, the phospholipid is a bilayer, so we have our polar heads pointing to the interstitial fluid and another polar head pointing to the intracellular fluid. Also, within this lipid bilayer, we have the cholesterol molecules. So I'm going to point to an individual cholesterol molecule right there. Cholesterol. You can see that it's found in between some of the fatty acid tails of the phospholipid bilayer, and it helps to secure the phospholipids together. It adds a little bit of stability to the cell membrane. So that forms the phospholipid bilayer, and the cholesterol forms the liquid portion of the fluid mosaic model of the cell membrane. Now we can see the proteins that are embedded in this liquid. The proteins act as the mosaic portion of the fluid mosaic model of the cell membrane. There are proteins that are integral. Right? If it's an integral protein, that means that it is literally a portion of the lipid bilayer. It could either extend halfway into the lipid bilayer, or it could go all the way through the phospholipid bilayer. If it does go all the way through or spans the entire lipid bilayer, we call it a transmembrane protein, and most proteins are transmembrane proteins. So here you can see one example of a transmembrane protein. You can see that that protein goes all the way through the phospholipid bilayer. However, there are some proteins that are peripheral. That means they're only found on the inner surface or outer surface, but do not actually go into the phospholipid bilayer. These are often attached to molecules that are integral or that are transmembrane. So here we can see an example of a peripheral protein. Now you may notice that many of these proteins have structures attached to them. For example, this protein right here has a structure that is attached to it. Those structures are carbohydrate groups. So many simple sugars or many monosaccharides bonded together, and they have these unusual three-dimensional shapes. For example, some of the carbohydrate portions could look like a Y. Some of them could have multiple prongs coming off of them, or even more branches. These are going to help in a number of ways. Sometimes they will provide sites for other molecules to attach. Sometimes the glycoproteins may indicate that this is one of your cells. We all have different glycoprotein structures that look different from each other. And as you can see, there's an incredible variety to this structure. So, let's look at some of the functions of the membrane proteins. Some of the integral proteins are ion channels. That means they will allow molecules to pass through them. Many of these are also transporters, so they selectively move substances through the membrane. So remember, Proteins often act as the gatekeepers, deciding what can enter a cell and what can exit a cell. Some of them are also receptors for cellular recognition. A ligand is a molecule that will bind to the receptor. So for example, proteins have often very complicated three-dimensional shapes. Here we can see a protein, and I'm going to draw an area on the protein. So that would be a protein. Because they have these complicated three-dimensional shapes, we can find other molecules that have the ability to fit those shapes and bind to them. So if we had a ligand shaped like this, like a little diamond, a ligand is a molecule that binds to a receptor. That ligand would be able to fit into that protein. So we would have a ligand binding to a protein, and that would cause a change to occur to the cell. Sometimes these proteins might be enzymes. Enzymes are proteins that speed up chemical reactions. They catalyze the chemical reactions. So if that little diamond-shaped ligand was actually a substrate in an enzyme-catalyzed reaction, you might have a transformation and a different product produced. Many of the membrane proteins may be cell identity markers. These are the ones that will allow the cells to identify cells belonging to you versus cells belonging to other individuals. In fact, there's a group of proteins with glycogen on them, so glycoproteins, that we call the major histocompatibility complexes. Major histocompatibility complexes, or we call these the MHCs for short. Now let's look at this term histocompatibility. Histology is the study of cells. A histology slide is a slide, a microscope slide, where you can look at the cells using a microscope. Compatibility means gets along with. So a histocompatibility complex would be a cell identity marker. They indicate the cells that get along with each other, all the cells of your own body. So here's a nice graphic that you can also find in your textbook that indicates the functions of these membrane proteins. Right? We can see that some act as ion channels. So we can see in these ion channels, ions or structures are allowed to flow into or out of the cell. Some will be carriers, so different types, often very large molecules, will be able to be moved into or out of the cell. These are often known as transporters. Receptors, and here you can see the ligand attaching to the shape of a protein. Many will be enzymes, so we have a substrate coming in and then a product coming out, products being produced. I didn't mention this one before, but sometimes these proteins will help link structures inside the cell to structures that are outside, often involved in linking two cells together. And finally, as I said before, the MHCs, these major histocompatibility proteins, often forms of glycoproteins that will allow you to distinguish cells that belong to you from cells that belong to someone else. So an important concept associated with the membrane is how do materials move into or out of the membrane. The cell is either permeable to certain substances or it is impermeable. Remember, the basic permeability is associated with the phospholipid bilayer. The phospholipid bilayer is a barrier to movement. The lipid bilayer, as we've already seen, is permeable to oxygen, carbon dioxide, and water, and other types of lipids, such as steroids. But it is impermeable to larger molecules that are somewhat polar, like glucose is somewhat polar. Since the lipid bilayer is a barrier, the cell has to have a mechanism for moving materials into and out of the cell, and that's the responsibility of the transmembrane proteins. They could be either a directly opened channel or a transporter that will allow certain materials like glucose or ions to pass through the membrane. So, movement of material into or out of the cell could either be a passive process or an active process. A passive process means that you don't need power from chemical energy. You don't need the power from ATP. So you do not need ATP to power these passive processes. They're simply using concentration gradients. So in a passive process, material is going to move down a concentration gradient. Active processes do require ATP. ATP is the common form of cellular energy. So a cell uses this energy from the breakdown of ATP to move material across the membrane, and usually it's going to be against a concentration gradient. So a passive process will move material down the gradient. An active process will move material against the gradient. Diffusion. Diffusion is the most common passive transport mechanism. Diffusion is simply the movement of material down the concentration gradient. So you can see in this illustration, at first you have a strong concentration of a dye molecule at the bottom of this graduated cylinder. So we have a high concentration at the bottom and a low concentration at the top. Soon, the dye particle will start moving generally down that concentration gradient until it is evenly distributed throughout the graduated cylinder. There are several characteristics that will determine how fast this diffusion occurs. The first is the steepness of the concentration gradient. So if you have a very shallow concentration gradient, material will diffuse slower. But if you have a very steep concentration gradient, material will diffuse much faster. Another determinant of diffusion is the temperature. Temperature is a measurement of the heat energy. So by increasing the temperature, you'll increase the general energy of the molecules. Remember, in diffusion, it is the kinetic energy of the molecules themselves that are powering diffusion. Also, the mass of the diffusing substance. The heavier the particle being diffusing through a material, the heavier it is, the slower. Next, the surface area. Now, this is where your cells in your body really can alter how fast diffusion will occur. For example, if you were looking at a cell lining your intestine, and it was just simply a cube-shaped cell, we had all sorts of particles getting ready to diffuse in. Those particles are limited. They are limited by this area, the area across which it will diffuse. However, if we change up that just a little bit, and let's say we add a cell with some villi on its surface, then if you look, you can see that the surface has a lot more area. So by increasing the surface area, you increase how fast the particles will diffuse across that membrane. And then also the diffusion distance. This just makes sense. If it has to diffuse farther, it's going to take a longer period of time before it comes to equilibrium. So there are all sorts of forms of diffusion involving the cell membrane. The first is known as simple diffusion. So in simple diffusion, a particle diffuses across the phospholipid bilayer. And remember, there's only a very small set of molecules that can diffuse directly across the phospholipid bilayer, including oxygen, carbon dioxide, and to a limited degree, water. Next, we have channel-mediated facilitated diffusion. So this diffusion is helped out by a transmembrane protein. That's where we get the term facilitated from. That means to help out. So you can see that in this facilitated diffusion, we have a channel, and items such as ions, for example, you might have sodium ions, cannot pass directly through the phospholipid bilayer, but if you have a channel that will allow positively charged ions, like cations, it will allow it to move through this channel. Finally, carrier-mediated diffusion. And in carrier-mediated diffusion, there is not a permanent channel through which the particle can flow. But anytime you add a structure to a protein, you will change the shape of the protein. So by adding this molecule to the protein, you will change the shape of the protein, may open up, and allow the molecule to leave on the interior of the cell. But in all of these, the particles are diffusing down their concentration gradient. Osmosis is a special form of diffusion. Osmosis is the net movement of water through a selectively permeable membrane from an area of higher water concentration to an area of lower water concentration. Right. Now, normally when we talk about concentration, we give the concentration in terms of the material dissolved in water. So, for example, I could put in a dividing membrane, and on one side of this dividing membrane, I might have a watery fluid containing sodium chloride, salt water, and let's say that this is 0.9% saline, sodium chloride. And on the other side, I put a solution that is 10% saline. So I'm giving you the concentration of the solute. But in osmosis, we're concerned with the movement of water through a selectively permeable membrane from an area of higher water concentration to an area of lower water concentration. So, which side of the membrane has a higher water concentration? That's right, the side that is 0.9% saline. We could essentially turn this number around on its head and say that this side of the membrane is 99.1% water. The other side of the membrane that has 10% saline we could essentially say that this was 90% water. It's always a good idea, if you're asked a word problem about osmosis, to draw it out. You often will be given the concentration of the solutes in the solution, for example, 0.9% saline or 10% saline. But if you're interested in which way the water goes, you have to figure out the relative water concentration. So always turn it around immediately to what the concentration of water is. Now we can easily see that this water can flow through the membrane down its concentration gradient. So the water will pass from one side to the other. It will pass from the area of 99.1% water to the area of 90% water. Water can pass through these plasma membranes in two ways. So far, I've only talked about it going through the lipid bilayer by simple diffusion. But remember, water is slightly polar. So it's slightly polar, but it's fairly small, so it can slip through the phospholipid bilayer, but not that well. There are also channels that will allow water to flow through fairly easily. These are known as aquaporins, which, as you can tell, literally means water pore, a water channel. So these are integral membrane proteins that allow water to pass through. There are several different types of aquaporins. When in A&P2 we talk about the urinary system, we're going to talk about a very important form of an aquaporin that allows you to produce either a very concentrated urine or a very dilute or watery urine. So osmosis is simply referring to the movement of water down its concentration gradient across a membrane. Well, the cell membrane is a membrane, so often we're interested in what happens to a cell when it's placed in solutions of different concentrations. This is known as the tonicity. Tonicity literally describes how a cell will behave. Now, let me give you a hint. Most cells on the face of the earth, including human cells, will have within it a relative concentration of 0.9% salinity. So often, salinity is used within osmosis problems. Right? Saline, a salinity, sodium chloride, when it is placed in water, will immediately dissociate into its two ions, the sodium ion and the chloride ion. These both have complete electrical charges. Sodium has a complete +1 electrical charge, and chloride has a complete -1 electrical charge. So these will not be able to pass through the phospholipid bilayer. There may be a few limited ion channels that will allow the sodium ion and the chloride ion to pass through, but there will be vastly more area for water to pass. So if you placed a cell, such as a red blood cell, in a different salinity of water, the primary movement is first going to be water. So if we place a red blood cell in an isotonic solution, and the term iso means the same, that means the concentration of the fluid outside of the cell is the same as the concentration of the fluid inside of the cell. Now, water will move into the cell, and water will move out of the cell, but it will move in and out at equal rates. Contrast that with what happens if you place that red blood cell in a hypotonic solution. In a hypotonic solution, that means the solution has less pull on water, so water will enter the cell. Now let's think about what's happening in terms of osmosis if you have a hypotonic solution. So if we have a cell, and remember, inside that cell is usually going to be 0.9% saline, and yet water begins to enter that cell, which side of the cell has the higher water concentration? Outside of the cell, the interstitial fluid, or inside the cell? And inside the cell would be the intracellular fluid. That's right. The interstitial fluid would have the higher water concentration. So that means that water will enter the cell. If it has a higher concentration of water, if you have a high water concentration, what does that mean in terms of the solutes? Yes, that means it has a lower concentration of the solutes. So in a hypotonic solution, water will enter the cell. Eventually, if enough water enters the cell could burst. In a red blood cell, this is known as hemolysis. 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 Next, we see the situation of a cell in a hypertonic solution. So in a hypertonic solution, we have a cell, and this cell is going to continually lose water. So there will be a net flow of water outside of the cell from the intracellular fluid to the extracellular fluid. That means that the outside of the cell has a lower water concentration. What does that mean in terms of the solute concentration? Yes, it will have a higher solute concentration. So water will move into the cell, water will move out of the cell, but the dominant direction will be out of the cell. And here you can see a red blood cell that is undergoing crenation. It is shriveling. Some people think that that looks like the tips of a crown, and a crown is crenulated. So far we've been talking about active transport. So far we've been talking about passive transport. Passive transport would require molecules to move down their concentration gradient with only the energy of the chemicals themselves. Active transport will use ATP energy and usually will move particles against a concentration gradient. So the solutes are going to be transported from an area of lower concentration of the solute to an area of higher concentration of the solute. A great example of this active transport is the sodium-potassium pump. Sodium and potassium are found as ions. So we have the sodium ion, and you can see that across the cell membrane between the extracellular fluid, the interstitial fluid, and the cytosol, the intracellular fluid, there is a gradient of sodium. Sodium is found in a higher concentration outside of the cell. So this outside of the cell has a high concentration of sodium ions. Remember, square brackets means concentration of. So we have a high sodium concentration in the extracellular fluid, a lower sodium concentration in the intracellular fluid. Potassium is just the opposite. There is a high potassium concentration inside the cell in the cytosol or the intracellular fluid, and a low potassium concentration in the interstitial fluid. So the sodium-potassium pump will pump using ATP energy, both sodium and potassium, and will pump them both against their concentration gradient. So for example, let's start off at step number 1. We see that we have a transmembrane protein that will allow 3 sodium ions to bind to it. Then in step 2, we add a little bit of energy from ATP that changes the shape of the protein, and the 3 sodiums are expelled to the extracellular fluid. Next, we've created an area for the transmembrane protein to pick up a couple of potassium ions. So in step 3, we have attachment of the 2 potassium ions. Then we expel the last phosphate group from the original ATP, and the potassium is shuttled or moved into the cell against the concentration gradient. And one thing I want you to see is that this is 2 potassiums. So we've been moving positive charges out of the cell and positive charges into the cell. Have we been moving equal number of particles out of the cell and into the cell? No. We've been moving out more positive charges and moving less positive charges into the cell. So not only are we creating a higher concentration gradient by moving the solutes against their concentration gradient, we've also created an unequal area of electricity. The outside of the cell becomes positively charged compared to the inside, which is negatively charged. And they're separated by a barrier. They're separated by the phospholipid bilayer. This is a battery. If you look at a battery, there's a positive pole and a negative pole. It's a form of potential energy. You get work out of it, you transform that into kinetic energy when you somehow attach the 2 poles. When you put it in a device that requires a battery, you end up attaching the 2 poles. You get kinetic energy. Well, that's what we're doing right here with this sodium-potassium pump. We're creating a battery out of a cell. And later on, we will allow those charges to rush back down their concentration gradient, and we will essentially connect the 2 poles of the battery and get some work out of it. We're going to see this in a lot of detail when we get to the muscle chapter and also when we get to the neuron chapter, because this is the primary mechanism that will help power muscle cells and neurons. The sodium-potassium pump is a very important concept. We pump out 3 sodium, 3 positive charges, and only pump in 2 potassium, 2 positive charges. It's unequal in the transport of the charges. Another way material could move across the membrane is by transport in vesicles. Vesicles are small intracellular, inside the cell, membrane sacs. This is considered a form of active transport because it requires cellular energy to manufacture more membrane material. Endocytosis is the movement of material into the cell, endo, inside, cyto, the cell. Movement of material into the cell in a vesicle formed from the plasma membrane. There are 3 primary types of endocytosis. The picture you see here is receptor-mediated endocytosis. And in receptor-mediated endocytosis, if we look at the cell membrane right here, you can see that there are receptors on the cell membrane that will bind a particle. When that particle binds to those receptors, it triggers the plasma membrane to circle around the particles and pull it into the cell. Later, the particles are broken apart by digestion with a lysosome containing lots of digestive enzymes, but the eventual goal is we've pulled particles into the cell using a receptor. So we call it receptor-mediated endocytosis. Sounds like a strange and complicated name, but really if you break it apart, each individual word makes sense. The next type of endocytosis, as we can see from this picture, is phagocytosis. And the term phago, P-H-A-G-O, literally means to eat. So this is going to be movement of a fairly large particle, like a microbe, like a bacterium, into the cell. The cell membrane will extend false arms called pseudopods out and surround the large particle and pull it into the cell. Then, similar to receptor-mediated endocytosis, a lysosome with its digestive enzymes will help digest the particle. Our final form of endocytosis is called pino or pinocytosis, and this literally means to drink. These are particles that are in solution. So the particles are dissolved, so they're in a solution. And a large section of this particle-rich solution is drawn into the cell. Once again, broken apart with a lysosome. Then we have exocytosis. We're only going to mention just exocytosis. In exocytosis, we have a cell, and you have a particle found inside a vesicle, or maybe a few particles inside the vesicle, inside the cell, and we will move that particle to the cell membrane surface and fuse it with the cell membrane surface and release the particles outside of the cell. If we coupled together endocytosis and exocytosis, then we could end up moving material all the way through a cell by having endocytosis on one side of the cell and exocytosis on the other side of the cell. All right, so we've talked a lot about the cell membrane. Next, let's talk a little bit about the cytoplasm and the two main components of the cytoplasm. First, we have the cytosol. That's the intracellular fluid. It surrounds the organelles. It helps suspend the organelles. There's lots of chemical reactions that occur here because remember, it's primarily made out of water, and water is really important for most metabolic reactions in the body. Many of these reactions will be to release energy. The energy from these reactions, and these would be, remember, exergonic reactions. Will be coupled together to provide the energy needed for putting molecules together, for putting building blocks together, adding amino acids together to form proteins, and this would be a form of, what kind of reaction would this be? An endergonic reaction. So we couple together the exergonic reactions and the endergonic reactions. Now, also found within the cytoplasm are the organelles. I've already talked about those a little bit. They have specialized structures and functions. They operate sort of like little organs inside the cell. We're going to go through many of the cellular organelles. beginning with the ones that are not surrounded by a membrane. One of the most important forms of these organelles include the cytoskeleton. The cytoskeleton is a network of protein filaments that span throughout the cytosol. It provides a lot of structural support for the cell, also helps move material around within the cell. There are 3 different types of these protein filaments. Beginning up at the top, the first type are the microfilaments. Microfilaments are often associated directly with the cell membrane. They help reinforce the cell membrane and help form the shape of the membrane or the shape of the cell. Here we can see on a special type of microscope, an inflorescent microscope, where cells are dyed so that if they are hit with a beam of light, they fluoresce. You can see the microfilaments in bright red or orange around the exterior portion of the cell. The second type include the intermediate filaments. The intermediate filaments are found all throughout the central portion of the cell and help stabilize the interior structure. They make the cytosol a little more jelly-like than watery. Finally, we have the microtubules. Microtubules are most associated with motion, and they literally do look like a little tube. We'll see them when we talk about cell division. They're going to be responsible for moving chromosomes around inside the cell. They're also responsible for moving other structures around within the cell. Many organelles will attach to microtubules, and the microtubules will move them around. Something else that the microtubules help form are very specific organelles of movement within cells. These include cilia and flagella. Cilia are short hair-like projections along the surface of the cell that move fluid along a cell's surface. Here you can see cilia that line the trachea. This is a scanning electron micrograph. Scanning electron micrographs are pictures taken with electrons bounced off the surface of a structure, where you can see a good 3-dimensional representation of the surface of a cell. Cilia are usually fairly short, and if we look at the movement of cilia over here, you can see that they move back and forth, sort of like oars on a rowboat. Next we have flagella, which are longer than cilia. They're responsible for moving the entire cell. The only example of a cell within humans that has a flagellum is the sperm cell. So here we can see the sperm with its very long flagellum. The movement of flagellum is a little more like a corkscrew. So if you look down in the lower right-hand corner, you can see the movement of the flagella, a little corkscrew propelling the cell forward. Continuing with our non-membrane bound organelles, again, organelles that don't have an internal membrane surrounding them, we have the ribosomes. Ribosomes are very important because that's where the proteins are manufactured. These ribosomes can be found free out in the cytoplasm of a cell, but often the ribosomes are going to be bound to a membrane. Please be sure to read in your book the section associated with construction of proteins on the ribosome. Now we're going to begin to look at organelles that are surrounded by membranes. The first of these is the endoplasmic reticulum. That's a very complicated term, but let's break it apart. Endo means inside. Plasmic is referring to the cytoplasm, so it's inside the cytoplasm, and it's a reticulum. A reticulum is a complicated network, a series of passages. And if you look at our endoplasmic reticulum right here, you can see that it is a complicated network of passages. We divide the endoplasmic reticulum into two forms. The rough endoplasmic reticulum, which is usually right up against the nucleus. The rough endoplasmic reticulum will have ribosomes studded on its surface. So here you can see all of these various ribosomes studded along the surface of the cell in the rough endoplasmic reticulum. Also, if you look down at the bottom, you can see some rough endoplasmic reticulum. This is with a picture taken with a transmission electron microscope. Transmission electron microscopes use a beam of electrons that are passed through a very thin section of tissue, and then you can analyze how easily the electrons pass through. It gives you very high magnification of a tissue section. So you can literally see the little dot-shaped ribosomes that are attached to the membrane, which appears as a line. Then we have the smooth endoplasmic reticulum. Smooth endoplasmic reticulum is located here. It's a little bit further away, but you'll notice there is often connections between the two. So it doesn't have ribosomes, so it's not going to manufacture proteins, but it will manufacture many of the lipids, including fatty acids and steroids, and many of those fatty acids will be important for detoxifying. Here, again, looking at our transmission electron micrograph, you can see that some of the endoplasmic reticulum does not have little dots or little ribosomes, so that would be the smooth endoplasmic reticulum. Next, we have our Golgi complex. So this is another membrane-bound organelle. Now, proteins, fatty acids are synthesized by the endoplasmic reticulum. The proteins, for example, in this picture at step number 1, proteins from the rough endoplasmic reticulum could be transported to the Golgi complex by some transport vesicles. The Golgi complex will pull in that transport vesicle by endocytosis, and the protein will be modified by the Golgi complex. So the Golgi complex will modify the proteins, sort them as to where they should go, and package them. Some people refer to this as a distribution center, a repackaging and redistribution center for the cell. Proteins are transported then out of the other side of the Golgi complex by various types of vesicles. For example, if you're only transferring vesicles from one area of the cell to another, we call them transfer vesicles. If you are transporting them to another organelle entirely, like to a lysosome, as in 9, we call them transport vesicles. If, as in number 7, you're producing something that will be secreted out of the cell, we call those secretory vesicles. And sometimes you make proteins that need to become a part of the cell membrane itself. We call those, as in number 8, the membrane vesicles. And you can see how the membrane fuses with the cell membrane, the plasma membrane, and leaves the protein embedded. We now have a transmembrane protein embedded in the cell membrane. So the cell has the ability to change the proteins that are found in the cell membrane. Lysosomes, another membrane-bound organelle that we've been mentioning a little bit so far. Lysosomes contain very powerful digestive enzymes. Why would you want to keep these digestive enzymes protected from the rest of the cell? That's right. If you just let the digestive enzymes loose inside the cell might digest itself and that wouldn't be good for cellular health. Mitochondria. Mitochondria are very important membrane-bound organelles. In fact, they have two membranes, an outer smooth membrane and an inner highly folded mitochondrial membrane. Mitochondria are often called the powerhouses of the cell, and they are responsible for generating most ATP energy. Now, not all ATP energy is generated in the mitochondria. There will be some that can be generated in the cytosol, in the cytoplasm. So most ATP energy that your cell creates is manufactured in the mitochondria. Some ATP will be found or be made out in the cytosol, the cytoplasm. So if you need a lot of ATP, like in a physiologically active cell, you probably will see more mitochondria. And when cells become increasingly active, you start seeing the cell self-replicating those mitochondria. For example, if you're a couch potato and you don't use your skeletal muscles that much, you're going to have the minimal amount of mitochondria in your skeletal muscles. But if you are an athlete, if you're very active and use your skeletal muscles often, then you will have more mitochondria in your skeletal muscles. Another really cool thing about mitochondria is that they have their own DNA. It's a form of a DNA molecule that looks very much like bacterial DNA. And these are inherited only from your mother. Sperm cells have very little cytoplasm and no organelles. Sperm cells are essentially a mass of DNA. So you would only inherit these mitochondria from your mother, so you often may hear something called maternal DNA. It's maternal mitochondrial DNA. And then we come to our last major section of the cell. Remember, the cell has three important sections: the cell membrane, the cytoplasm, and the nucleus. So the nucleus is a very large structure, very important for controlling the activity of the cell. Nuclei may be spherical or they might be oval shaped. Usually, if you're looking at a light microscope slide, it may be one of the only structures you can physically see inside the cell. The nucleus is surrounded by some membranes. We call it the nuclear envelope because it's a double membrane. Two phospholipid bilayers that separate the nucleus from the cytoplasm. The nucleus needs to protect your DNA from anything that might happen to it. Cytoplasm is usually very metabolically active, and the DNA might become damaged. So most of the time your DNA is kept inside the nucleus, protected from any chemical reactions that are happening in the cytoplasm. The nuclear envelope has nuclear pores because sometimes you do need material to pass into or out of the nucleus. But these nuclear pores are guarded. There are cells that surround the nuclear, not cells, I'm sorry. There are molecules that surround the nuclear pores that will control what can enter or exit the nucleus. The nucleolus is a structure inside the nucleus where most of the ribosomes are manufactured. So the ribosomes are manufactured in the nucleus, and then they pass out of the nucleus to the rest of the cell. So a really important thing that the nucleus contains will be your chromosomes, those long molecules of DNA combined with protein molecules. So how many molecules of DNA does a human have? That's right, 46 total. How many pairs do we have? That's right, 23 pair. On those chromosomes, you'll find the genes, the cell's hereditary units. So these are sections of the chromosomes that will code for a structure, and the structure that they will code for will be proteins. So genes actually code for proteins. And remember, a really important function of many proteins is as an enzyme. And enzymes control all chemical reactions that occur in the cell. All those chemical reactions will be sped up by enzymes. If you don't have the right enzyme, the chemical reaction does not occur. So in this way, your DNA controls the cell indirectly by control through enzymes. So here we can see a picture of a nucleus. If you look carefully, you can see the nucleus with the rough endoplasmic reticulum attached to it from the outside. We can see the double membrane, so when you look at the details of the nuclear envelope, you can see the double membrane with the very well guarded nuclear pore, allowing some material to pass in and out. We can see the nucleolus right here, but here's a word we haven't seen before. It's called chromatin. Now remember, way at the beginning of our discussion of the cell, I said that sometimes when we use the word chromosome, we are simply referring to the molecule of DNA with its associated proteins, but sometimes when we use the word chromosome, we're talking about the highly coiled up, condensed shape of a DNA molecule that you only find during cell division. At times other than cell division, the form of the DNA is as long, thin strands, and we call that chromatin. So chromatin will be the long, thin strands of DNA that will be found when the cell is not replicating, when the cell is not dividing, when it's not going through mitosis. So here we can see how the DNA can be packaged into a chromosome of a dividing cell. So based on that phrase, is this chromosome talking about simply the molecule of DNA or the specific structural form of DNA that you see when the cell is dividing? That's right. It's talking about the structural form of DNA that you see when the cell is actually dividing. The chromatin is a relatively loosely packed version of the DNA that you see when the cell is not dividing. So you can see that the DNA wraps itself around proteins and then coils and coils again until we have a chromosome. Now this chromosome is interesting. So this chromosome that we see here is interesting in that it is actually made up of two identical DNA molecules attached together at an area we call the centromere. Right? During the process of the cell cycle, the DNA at one point will make an exact copy of itself. That's called replication. So in the process of replication, the cell, the DNA makes a copy. So we start off with one molecule of DNA and we end up with two molecules of DNA of the same chromosome. Those two molecules of DNA remain attached to each other at this structure we call the centromere. So when the DNA coils up to form the structure we call a chromosome, it's going to have two portions to it. Each of those portions is called a chromatid. So you can see that this DNA molecule, this chromosome, has two chromatids. And in the process of cell division, eventually it will separate at the centromere, and then one newly formed cell will have one chromatid, one newly formed cell will have the other chromatid. So let's go ahead and talk a little bit about somatic cell division. Now, first off, let's hit this term somatic. We're going to use this term a lot in the future. The term somatic literally means body. So this is body cell division. All cells in your body other than your sex cells that are found in your testes or your ovaries. So the cell cycle is a sequence of events where a body cell, a somatic cell, can duplicate its contents. So it will duplicate the DNA, it will make a lot more organelles, and then divide into two. So human body cells, human somatic cells, have 23 pairs of chromosomes for a total of 46. Now from that phrase, I'm really simply meaning the DNA molecules. So a human somatic cell contains 46 total DNA molecules and their associated proteins. The two chromosomes that make up each member of a pair, so remember they're paired up, are called homologous chromosomes. Homologous chromosomes contain similar genetic information, but it doesn't have to be identical. For example, my father has brown eyes and my mother has blue eyes. So I may have gotten one bit of genetic information from my mom that says blue eyes, and that's contained on one chromosome. My father may have given me another piece of genetic information on another chromosome that says brown eyes. Now both of them have genes that are all about eye color, but they don't have to be exactly the same. So we call those two chromosomes that are paired up homologous chromosomes. So somatic cells contain two sets of chromosomes, so we call them diploid cells. Diploid cells. In the cell cycle, we have two main phases, interphase and the mitotic phase. So we have interphase and the mitotic phase. During interphase, the cell is, well, it's going to do its normal day-to-day operations, but it's also going to replicate its DNA. So there are three main phases to interphase, and this literally means the phase in between cell division. There's G1, S, and G2. During G1 phase, the cell is going to grow. It's just divided, so it's going to grow a little bit. It will make decisions about whether it should continue with the cell cycle and go ahead and replicate the DNA, or whether it should exit from the cell cycle and become a non-dividing cell. If it does decide to exit from the cell cycle at this point, we call that G0 phase. Then the cell goes into S phase of interphase, and that literally stands for synthesis, making the DNA. So we replicate the DNA, we make an exact copy of the DNA. So after that point, each chromosome has two DNA molecules. Then we go into G2 phase, where the cell will essentially get ready for cell division, but will continue to grow. It'll manufacture some extra organelles and get ready for cell division. And then we enter into the mitotic phase. The mitotic phase has two major events. Mitosis itself, which is dividing the nucleus, and dividing the cytoplasm of the cell. We call that cytokinesis. So by the end of the mitotic phase, we have finished both mitosis and cytokinesis, and we have two small new cells that then go directly into G1 of interphase. So the DNA is going to make an exact copy of itself. And if you've read about DNA in the book, or if we've talked about it in class already, you can see that the DNA is a double-stranded molecule. On each of the strands, we can see various nitrogen-containing bases. So these over here are nitrogen bases. The 4 different nitrogen bases of DNA include adenine, guanine, thymine, and cytosine. And these bases pair up in what we call complementary base pairing. Adenines will always match up with thymines, and guanines will always base pair or match up with cytosines. So during DNA replication, the 2 strands of DNA separate and the complementary base pairs are added in, therefore creating 2 new double-stranded DNA molecules, each of which are made out of one of the old strands and one of the new strands. Now let's talk about mitosis. Mitosis is divided into 4 main phases. Prophase is the phase in which the chromatin fibers, the long thin strands of DNA, physically condense into the chromosomes. Usually the nuclear envelope also breaks apart, and you can at this point physically see the chromosomes, but they'll be masked up in the area where the nucleus used to be. Also during prophase, a structure called the spindle begins to form. The spindle is made out of microtubules which will attach to the chromosomes and move them to the center of the cell, which leads us to our next major phase of mitosis, metaphase. In metaphase, the microtubules are aligned along the center of the cell, the metaphase plate, by the centromeres. So the chromatid pairs line up along the metaphase plate, being pulled there by the microtubules of the spindle. During anaphase, the chromatid pairs split apart. So we would have started off with a duplicated chromosome made of 1 and then 2 chromatids. And in the process of anaphase, those 2 will split away from each other and begin to move towards the opposite sides of the cell. Telophase is essentially the opposite of prophase. The DNA is now at the 2 ends of the cell, and new nuclei begin to reform. The DNA relaxes from the chromosome form back to the chromatin form, and the structure of the nucleus begins to reform. Now remember, we also have to accomplish cytokinesis, and this will overlap with mitosis. It's not mitosis then cytokinesis. Mitosis and cytokinesis overlap with each other, but this is going to divide up the cell's cytoplasm. Remember, during the last phase of growth in interphase, the cell made a lot of new organelles. So the newly formed cells would have some of each of the organelles. So this usually begins in late anaphase with the cell elongating a little bit, and then the plasma membrane will begin to be pinched along the middle, forming a structure called a cleavage furrow, which will keep getting deeper and deeper until the cell eventually will split into 2 daughter cells. Usually the cleavage furrow is very deep as we get into telophase. Interphase then will begin when cytokinesis is complete. So here we can see the phases of mitosis, beginning up at the top with 1, which may be a little bit off of this video, but you can still see that it is interphase. The cell is perfectly formed. Then we go into 2, which are 2 views of prophase. You can see that you can distinctly see the chromosomes. They're masked where the nucleus used to be, but you can see the fiber structure of the chromosomes. Then the spindle will pull the chromosomes to the center of the cell in 3, where you can see all the chromosomes lined up along the center of the cell. That's metaphase. And then we go into 4, which is anaphase. So the cell elongates a little bit, and you can see on the right side of 4, it looks like the chromosomes are just a little bit separated. And then on the left side of 4, that's a classic looking anaphase, where you can see the 2 sets of DNA heading towards the opposite sides of the cell. And then as we move up into 5, the DNA is at the 2 poles. You're going to begin to reform the nucleus, and cytokinesis is almost complete, but you can still see there's a little bit of a connection of some fibers in between the newly forming cells until we get up to 6, where we have 2 completely newly formed cells, each with a distinct nucleus and surrounded by an intact cell membrane. Now, so that was somatic cell division, body cell division, but we also need to divide the cells for reproduction. So during sexual reproduction, each new organism is formed from union of gametes, sperm or egg, from each parent. That's fertilization. So, the sperm and the egg are formed through a type of cell division called meiosis. It occurs only in the gonads and produces the gametes, the sperm or egg. The key about meiosis, however, is that we have to reduce the total number of chromosomes. If we didn't, then you would have a sperm from the father containing 46 chromosomes combining with an egg from the mother containing 46 chromosomes, and the offspring would have 92 chromosomes, and that's way too many. So in the process of meiosis, we've got to reduce that 46 down to 23, exactly half in the male, and 23, exactly half, coming from the female, and that's going to give us a resulting final cell that has 46 total chromosomes. We call that halving of the number of chromosomes formation of a haploid cell. So those would be gametes that contain a single set, and for humans that would be a single set of 23 chromosomes. Fertilization, union of sperm and egg, will restore the diploid number of chromosomes, 46, 2 times 23. Reproductive cell division looks a lot like mitosis. However, it occurs in 2 stages. Now, prior to meiosis, the cell is going to go through interphase. And remember, in interphase, the DNA makes an exact copy of itself. So, each of these phases of meiosis has 4 phases: prophase, metaphase, anaphase, and telophase. So if it's occurring during meiosis 1, we call it prophase 1, metaphase 1, anaphase 1, and telophase 1. If it's occurring during meiosis 2, we would call them prophase 1. If it's occurring during meiosis 2, we'd call it prophase 2, metaphase 2, anaphase 2, and telophase 2. So just a brief summary of reproductive cell division. We begin meiosis 1, we have a diploid cell, but remember, the cell has gone through interphase, so each chromosome is made out of 2 DNA molecules. We end up, after meiosis 1, with 2 cells having the haploid number of chromosomes. So in the process of meiosis 1, we separate the homologous pairs of chromosomes. In meiosis 2, we start off with those 2 haploid cells, but remember, each of those chromosomes still has 2 DNA molecules. So in meiosis 2, we separate the chromatids. Meiosis 2 actually looks nearly identical to mitosis. The net result will be 4 haploid gametes that are all genetically different. It's a reshuffling of the chromosomes from each parent, genetically different from the original diploid starting cell. So here's a brief overview of meiosis and then cytokinesis associated with meiosis. So we start off with duplicated chromosomes. In prophase 1, those duplicated chromosomes, each of them having 2 chromatids, pair up in their homologous pairs. In the process of meiosis 1, the homologous pairs will separate. So the homologous pairs line up in metaphase 1. The homologous pairs separate in anaphase 1, and we begin to reform the nuclei in telophase 1, and we divide the cells. Then we begin with meiosis 2. So we have our 2 cells beginning meiosis 2. In prophase 1, the chromosomes recondense, so you get those distinct structures we call chromosomes. Each individual chromosome lines up along the center in metaphase 2. In anaphase 2, the sister chromatids separate from each other, and in telophase 2, we reform our nuclei, each of which are now haploid. So we went to the haploid number at the end of meiosis 1. So we have 4 haploid cells. So here you can see a side-by-side comparison of mitosis and meiosis. The only difference is in meiosis 1. Meiosis 1 separates the homologous pairs. Meiosis 2, along with mitosis, separates the sister chromatids. So, we've been talking a lot about cells, and there are a lot of different types of cells. The average adult has nearly 100 trillion cells and about 200 different types. So you have muscle cells and nerve cells and blood cells, epithelial cells and connective tissue cells. There's a lot of different shapes and sizes of cells, but we all start off as a single-celled organism. So that single-celled organism will undergo cell division and differentiation, which literally means that the cells become different. The diversity of cells that we have allow us to form very complicated tissues, each with specific structures, and have various combinations of those tissues into organs, which allow our body to operate very efficiently. And this concludes our discussion of cell biology. Please watch this video again at your leisure, take additional notes, be sure and read the book and compare the notes from the book and the online lecture. Look to Wiley Plus for any available homework assignments, and do any quizzes as they become due. Thank you very much.